Hi everyone, I am Esra Yıldırım Sirkeci. I am a master's student in Bioengineering department at Marmara University. Today I'm going to present my master thesis, which is network-based integration of multi-omic profiles to purpose new biomarker candidates in pancreatic cancers under the supervision of Dr. Beste Turanlı. The content of my presentation will start with the brief introduction of the context of my work, then the aim of the study, followed by study design used in this research and achieved with the results and a conclusion. Pancreatic cancer is an aggressive cancer of the digestive system with increasing incidence and very high mortality. To date, surgery is the only treatment option available. However, the 5-year overall survival time remains unsatisfactory. Late detection and the particularly aggressive biology are the major challenges which determine therapeutic failure. Most patients with pancreatic cancer are diagnosed at advanced stages when curative surgery is no longer possible. Accordingly, outcomes for patients with pancreatic cancer are always poor. Robust biomarkers with high sensitivity and specificity for early detection will enable curative resection of pancreatic cancer, reducing the high mortality rate. Therefore, the development of early diagnostic pancreatic cancer biomarkers is an urgent clinical concern. Systems biology studies of pancreatic cancer rely on the integration of omics data from different biological levels. Systems biology provides a powerful tool for understanding diseases and solving challenging biological problems, allowing biomarkers to be identified and quantified with increasing accuracy and sensitivity. With the frequently arising challenges regarding cancer diagnosis and treatment, the holistic view provided by the systems biology approach allowed for having a global understanding of the mechanisms of the disease and gaining more insight toward diagnostic or prognostic biomarkers and drug target discovery. So this perspective provides a crucial network to integrate multi-omics approach to biomarker identification, shedding additional light on early pancreatic cancer detection. Circular RNA is a special type of endogenous non-coding RNA formed by reverse splicing of exon or intron cyclization. Recent studies have found that circRNAs are associated with many types of cancer, and several circRNAs have been identified as novel cancer biomarkers. CircRNAs are members of the competing endogenous RNA family and act as regulators of microRNA activity. They can competitively inhibit the binding ability of microRNA and their mRNA targets. In recent years, increasing evidence shows that circular RNA disorder is closely related to tumorigenesis and cancer progression. However, the regulatory functions of most circRNAs in PDAC remain unclear. So this study was aimed at exploring the molecular regulatory mechanism of circRNAs in PDAC, and the findings of this study might provide a deep understanding of the circRNA-related competitive endogenous RNA regulatory mechanism in PDAC pathogenesis. Here you can see the study design of these theses. First of all, we did data collection from Gene Expression Omnibus. Then we found differentially expressed genes. After that, using these differentially expressed genes, we construct PPI, protein-protein interaction network. So after that, we construct 3 key modules using ENCODE plugin. Then functional enrichment analysis was performed, and finally competing endogenous RNA network was constructed successfully. You can see the list of mRNA, microRNA, and circRNA study samples from Gene Expression Omnibus in order to identify differentially expressed genes. 2 circRNA expression profiles, 3 microRNA expression profiles, and 4 mRNA expression profiles were downloaded from GeoDatabase. In this study, 248 differentially expressed genes identified by analyzing 4 sets of mRNA expression profiles and 10 differentially expressed microRNAs from 3 microRNA expression profiles, and also 110 differentially expressed circRNA identified from 2 circRNA expression profiles. After the analysis with the GO2R tool, we set the cutoff point as an adjusted p-value less than 0.05, and the direction of gene expression was determined due to the log FC less than 0 as downregulated or more than 0 as upregulated. Then the mRNAs, microRNAs, and circRNAs were ranked by the absolute value of log FC. And here you can see the heat map analysis of differentially expressed mRNAs, microRNAs, and circRNAs. Heat map of the upregulated and downregulated genes, which are common between datasets in the A and C, and the heat map of common differentially expressed microRNA in the B according to the log FC values. After that, functional enrichment analysis and pathway enrichment analysis were performed to explore the roles of these differentially expressed genes. And the top 10 enriched GO terms shows that the differentially expressed genes were involved in regulation of cell adhesion, degradation of extracellular matrix, regulation of muscle adaptation, protein phosphorylation, and regulation of kinase activity. 248 differentially expressed genes were used to establish the protein-protein interaction network using the Cytoscape software. And the protein-protein interaction network consisted of 1,700 nodes and 2,136 edges. The visualization and topological analysis of PPI network were carried out through Cytoscape, and the dual metric approach, degree as a local metric and betweenness centrality as a global metric, was applied to the PPI network of each dataset to determine hub protein using CytoHubba plugin. And here you can see the top 20 hub genes with their betweenness centrality and degree values, and the redder the color, the higher the ranking. And based on the degree of importance, 3 key modules were then screened from the PPI network using the ENCODE plugin, and pathway enrichment analysis was performed. Module 1 contains 20 DEX, Module 2 6 DEX, and Module 3 4 DEX. The color of each node represent differentially expressed genes, and the results indicated that 8 of the 20 hub differentially expressed genes, such as CDK19, ACT, and 4TP53 in the PPI network, were distributed in the 3 modules, suggesting that these genes may have important roles in pancreatic cancer. For example, TP53 is one of the most frequently mutated genes in all cancers and is mutated in 70% of pancreatic cancers. And for ACTN4, in a study, they have identified increased expression and gene amplification of actinin-4 in pancreatic ductal adenocarcinoma and clarified its clinical and biological significance. And the competing endogenous RNA regulatory network was constructed by combining circRNA microRNA pairs with microRNA mRNA pairs. And the network contained 56 circRNA microRNA pairs and the 41 microRNA mRNA pairs, including 46 circRNA, 10 microRNA, and 31 mRNAs. And the present study successfully established a circRNA-associated ceRNA network via bioinformatics analysis. And the results shows that a circRNA 000554, which has most interactions, may play important roles in PDAC progression, which provides new insight into the pathogenesis for patients with PDAC. Furthermore, the conclusions of the present study are only based on the current existing tools and databases, thus it can be supported by further validation experiments. So, thank you for listening.